So this is what I feel. These are the difficult areas in botany. What are the difficult areas in botany? A very important chapter, plant kingdom in the first year, where most of you feel that the life cycle part and various examples, and even which plant has a dioecious nature, monoecious nature, and which is heterosporous, homosporous. All these things, these are the different areas which you feel somewhat difficult. Even in a morphology, you don't find correct examples to be memorized. And even in definitions, you don't find to remember them. For example, a definition for the Nistidi situation or definition of a parthenocarpic food. And sometimes recently also when a set of the NEET pattern of questions is 4 or 5 examples are supposed to be listed, study the following, and a list of 7 or 8 plants will be given, and how many of them are belonging to which family or how many of them are medicinally important. If they are asking, so number wise, you are not able to segregate them because you don't remember the economic importance of those plants correct. That's also a point. And very difficult topic for most of the students, the anatomy is the secondary growth. That's very, very important. A number of times they go on studying, but they find it difficult because which cambium is formed by differentiation, which is formed by dedifferentiation. These things they find it somewhat difficult. So this is also one difficult area. And in cell cycle, cell division, exactly what I have written there is the heart of the chapter. Subphases of prophase 1 and the major events which differentiate between mitosis and meiosis. Mitosis and meiosis. These are the two important difficult areas in the aspect of cell cycle, cell division. Coming to the photosynthesis, the chemiosmotic theory, which is discussed along with non-cyclic electron transport. And most of the students recently also, they failed to answer a simple question, which is the primary CO2 acceptor in C3 plants. Then automatically they went on keeping Rubisco. It's not Rubisco, it is RUBP. So here you need to have a clarity over what the question is, whether it is on the primary acceptor or the enzyme, what they are asking. So that important point, that's why I presented there C3 cycle and C4 cycle. Then the respiration part, that is electron transport chain. It is also like secondary growth and the PCA cycle, which is a substrate level phosphorylation reaction where GTP comes out and where ATP comes out, and also the ATP calculations. For example, from pyruvic acids, how many ATP you will get? 15, 14, 18. What's the answer? So that part also, most of the students they find it difficult. So you need to have conceptual nature here. And in PCA cycle, you will be able to notice that which is a mobile carrier, which is not a mobile carrier. That part, the number of complexes, how are the electrons and protons moving, all this thing goes to be somewhat difficult for the students. That's also an area where more conceptual knowledge should be there. The next one. So this is regarding the second year syllabus, where microsporogenesis, megasporogenesis, even which are endospermic seeds, non-endospermic seeds, what supposed to be a parthenocarpic fruit, what is a true fruit, what is a false fruit, and what's the ploidy levels of various tissues, that is endosperm of gymnosperms, endosperm of angiosperms, what about the ploidy of the nucellus? These are the areas where the student needs to have more clarity and they regularly get confused. So this is on the sexual reproduction part. And as I told you while presenting you the slides itself in molecular biology, the regular part is replication, translation, lack of conceptually the methodology, which step occurs after which step in translation, because the textbook has a limited explanation of half a page, but the questions will come from that half a page more in an applicative manner or in a direct. And by technology, you observe that the procedure of recombinant DNA technology, especially the selection of recombinants, that is when you place a desired gene in the antibiotic resistant gene, that is a selectable marker, which will help in the selection of recombinants, which will help in the selection of transponders. That's also a point which you need to understand. And in genetics, there will be a table which differentiates the genes and chromosomes. It starts with chromosomal theory of inheritance, followed by what in the sense linkage. And the NCRT people presented a big single page explanation in the form of a diagram for the linkage cross A and cross B with regard to Drosophila work of T.H. Morgan, where most of the students find it difficult. So linkage is also a very difficult topic. This is on the class 12 part.